Dr. Rowe for Venerology Doc, drrowe.com, and neurokc.com, all arms of the Rowe Neurology Institute to promote education and neurologic disease. So today we're going to talk a little bit more about how drugs are developed. They start with an idea, but an idea has to have legs, and an idea is not patentable, and any drug without patent protection cannot be developed, cannot make it through the hoops that it needs to develop to get out to patients. That's the brutal truth about drug development. But it's what we're dealing with, and it's a way hope can be given to patients, and the way therapies can be developed to improve patient quality of life in the absence of a cure and the absence of a known cause. And we deal with this all the time in neurology. So first of all, start the idea, then the initial work in the lab, side by side with PhD investigators. I know this because I've participated in 150 clinical trials, and I've actually come up with a way to make X-ray contrast, say for the kidney, when I was looking for something else, and I'm a physician scientist. Then a startup is formed, and the startup and it usually involves angel investors who believe in the technology and who are savvy enough to know that there's a very good chance the startup will fail. 95% of startup companies fail. They fail for various reasons, but the major one is management and a lack of clear goals for the startup vehicle. And these companies fail in the $1 to $1.5 million range, and they sometimes venture capitalists get involved in these. Sometimes large pharmaceutical companies get involved in repurposing a drug. But the valley of death for startups is in the $1 to $1.5 million range. Beyond that, though, if you're lucky, and if you're savvy, and if your concept is valid and you have patent protection, then you can sell to an intermediate pharma company. The intermediate pharma company then goes to the FDA and has a pre-IND meeting, pre-investigational new drug meeting. And the FDA very carefully specifies what needs to be done in the development. Is that drug safe enough to go into humans with preclinical studies? And then once it is determined to be safe enough, then phase 1, 2, and 3 studies, and then post-marketing studies are carried out with that particular drug. Phase 1 is generally a safety study. Are there signals that the drug is unsafe coming out in the first human volunteers? Phase 2 studies are generally dose-finding studies. If the drug is safe, then they have to, but the actual dose of the drug has to be determined. Then the final study is the phase 3 study, or the randomized double-blind placebo-controlled study that actually shows a drug is either safe or not, is either effective or not. And then there's marketing and FDA approval. So this is a very long process. But to have drugs that come onto the market that are safe and actually work to improve the quality of life of patients and to treat disease, it's necessary to go through this rigorous process. Devices, on the other hand, are much easier to get approved through a predicate process, but we're not dealing with that today. So don't be afraid of FDA monitored clinical trials, because they've gone through a lot of initial work before they'll ever make it to use.